introduction of the working group 3 by Jan and free presentation after.
Thank you very much, Benoit. My name is Jan Jansa, and I'm alone taking care of this work group. So if there was interest for a co-chair, especially with some expertise in economy, in putting dollar signs of the ecology services or ecosystem services, that would be really great, because that's something what we are looking at and what we don't have yet that much expertise. Right. So the main objectives of the work group 3 is to establish the network of researchers. This is what we're doing. Also to support their applications for funding. And then the main core of the activities should be to provide a list and to assess and quantify the benefits by the beneficial microbes with from various perspectives. From the nutritional, non-nutritional point of view, from interaction with potential or realized pathogens, environmental stability, various kinds of stresses, and also mitigation of risks and potential for battling or combating the water and air pollution. And then it's putting the dollar signs of those. That's the most challenging one. To know, okay, is this worth it? Is this competitive with the fertilizers price, for example? So that's the idea behind all this. And I wanted to go briefly through the different things. So the benefits, there are many benefits. There would probably not be thousands, but there would be dozens of benefits. And I think no one is thinking really in the broader scope. So that's really where we have to get together. At least in the microbial field where we are now, there has been for a long time, people were obsessed with nutritional benefits. But now there's more and more indication that the mycorrhiza is very important also for disease resistance. This is not that elaborated, so all these biotic stresses. People have worked on this, but this has never attracted so much attention. So we know there's a lot of microbes, we know there are potential pathogens, and they can also, their classification may be a little bit fluid. So that will be one of the specific objectives to get a list of benefits. And the second one is to put the economic values, taking into account also the material costs or the life cycle analysis, where we would also account for the resources that might be to some extent also not really recyclable. The other point or the other scope is to look at the value for the industry or enterprises, and then also to look at the functional diversity, at the synergies or antagonism within communities. And how these benefits are potentially modulated when the environment is changing. So everything is moving now towards differences from what we have been used to in the past, and so will the benefits if or the interactions that are present in the systems. So also to look from the angle of the global changes, and. Of course, together with the other work group packages, particularly the last one, we should also be thinking how to secure the agricultural production base in a system which is stable, which is almost not happening, or changing or fluctuating, because there might be also repercussions of the system. It doesn't have to be a monotonous change, but it can also be a variable landscape. And this is not only climatic, but we also put on purpose that we are talking about geopolitical factors, because there are things that might actually affect the food base or global food base or global food reserves, just because there are geopolitical issues. And so that's about the objectives. And we have proposed 6 deliverables to be contributed to by this working group. And the imminent one is providing a list of services conferred to agro ecosystem and the human society. So here there's a clear aspect we are not only talking about whatever function in the ecosystem, but this is a contribution to the human manipulated or human managed ecosystems, and not only to provide a list, but also to do some brainstorming how to measure them, whether to measure them directly or to use some proxies, and how these proxies relate to the reality. And there are 3 more, which only come in a more distant future, which should be also more elaborated in a form of publication, and they will be working on these services that should be identified relatively soon, and they should analyze how these services link to the beneficial interaction networks. Then we hope to also provide some concepts, and possibly also based on some theoretical modeling, how the economy benefits could change in the changing world. And another one, which also should be either report or publication, which is evaluation of the beneficial interaction networks with respect to ecosystem resistance and resilience to changes. There is almost 150 participants that actually showed their interest in participation in this working group from many countries, including some of the more distant ones. But of course, there's a lot of overlap with the other working groups. So there's almost no one who has an interest in just single working group. So this will be also a challenge how to split tomorrow for the specific discussions into the working groups. With this, I would like to end and invite you for the brainstorming about how to achieve the goals of the working group 3 tomorrow in a room that I don't remember. But I think we will get the information in time. Thank you very much.
So we have 3 presentations now. The first one is from Sofia Pereira.
Hello everyone, good afternoon. First of all, I'd like to thank the opportunity to be here today and share the data that we have obtained regarding the use of bioinoculants and biochar, their effects on grapevine-associated soil microbial communities. So I'm Sofia Pereira, and I'm a senior researcher and also invited assistant professor at Catholic University in Porto, in Portugal. So a lovely city, you are all welcome. And today I'm going to show part of the work that we have done under the scope of the PRIMA project, Recrop. So Recrop arose from the need to find solutions to improve the sustainability and the resilience of the Mediterranean farming systems. As you know, they are being negatively impacted by the climate change and also by the use of non-sustainable farming practices. So the main objective of this project was to foster the sustainability and also the resilience of these agricultural production systems by combining the use of biotechnological tools, in particular the use of bioinoculants with mycorrhiza, plant growth promoting bacteria, single or in combination, and also combine this with the use of environmental friendly agronomical practices. And here I'm talking about the use of organic amendments, for instance, instead, in order to try to reduce the chemical fertilization, intercropping schemes, and so on. So the Recrop consortium has involved 7 countries. Portugal is the coordinator, but we also have partners from Spain, France, Italy, Morocco, Egypt, and Tunisia. And as you can see here, we have a vast edapoclimatic condition. So we are trying to put these solutions in practices in a wide range of climatic conditions. So the strategy of RECROP is based on the evaluation of these beneficial effects of the bioinoculants, cropping patterns, and also soil amendments in three important, in terms of economics, Mediterranean crops, so the vineyards, the maize, and also the aromatic and medicinal plants. And we also have here a multi-actor approach. So because we also have a close connection, each country has a close connection with farmers and farmers associations in order to find the main barriers, and also how they see these kind of solutions as a good solution to be adopted in the future. We have dedicated partners in the socio-economic aspects related to the project, our French partners. They are trying to developing the participatory research actions and also models to compensate the farmers in order to use these greener solutions if they have any kind of losses in the production. So the main expected impacts of the project, of course, are to increase the soil health, the fertility, and also namely the fertility and the soil functions, and increase the resilience of these agroecosystems by better adaptation to climate change, and of course here the social and the economic development. But the project also has a huge focus on soil biodiversity. So by the end of the project, anyway, we are halfway of the end of the project. We want to have a comprehensive characterization of soil biodiversity profiles through the combination of classical taxonomy, because we also are evaluating the mesofauna and macrofauna, not only the microbiota. That's why we have here the classical taxonomy and NGS technologies, and in order to promote this productivity biodiversity binomial in order to help farmers to in the decision making. So in particular case of this work that I'm going to present, we use two different soil amendments. So we use the biochar and also a water retention, in particular case the hydrogel, in order to improve the water retention in the soil, which is one of the main focus of the project. So we have in the consortium at least 16 already established field trials that have been monitoring and where we have applied different types of strategies regarding considering the best that the best fits the interests of each country and crop. And in Portugal, in particular, we have a field trial established in the Douro region, which is one of the main or the most important regions of wine production. In this field trial, it was established in 2017 under the scope of another project that we have been monitoring these results. So the strategy here was to apply different microbial treatments. So we have PGPB single inoculated, and then also EMFs, commercial EMF, and also a mix of these treatments. And we have monitoring this field trial not only in terms of soil microbiota, but also in terms of plant productivity, must attributes, and so on. And in 2022, we established a new treatment in this field trial where we have applied cover crops in the intervals of the vineyards in order to try to increase the water retention in this area and try to help the grapevines. So our first sampling regarding the microbial analysis was in May 2022, and I have here some preliminary results, very, very preliminary results regarding the bacterial communities. As you can see here, we did not find any kind of effect when we applied microbial treatments, both in terms of composition of the communities, but also in terms of ecological functions. But actually, we are still, of course, analyzing all the data that we have available. But I would like to share with you that besides we don't have these effects in microbial communities, we had very interesting results regarding the must attributes. So in the treatments where we have microbial inoculation, we observed an increase in the fructose and glucose So the sugars that are present in the grapes, and also in terms of organic acids, we have high increases in the succinic acid, for instance, in tartaric acid, and also a huge decrease in citric acid. We have applied the same microbial inoculants in another vineyard in Spain, and we also had the same results. So we are trying to understand with the winemaker how this could impact the quality of the wine. And we also have another field trial in Vinho Verde region, also in the north of Portugal. And here the strategy was a little bit different. So we have also used microbial inoculation and also biochar. But in this particular case, we grown the grapevines in pots with and without biochar. And we have inoculated them with PGPVs and EMFs. And after 1 year, these grapevines were re-inoculated and transplanted to the field trials in Quinta do Azevedo. And after 2 years, we have collected some samples for microbial diversity analysis. And regarding the bacterial communities results, we saw that the treatments, both biochar and microbial inoculants, did not have any impact in the alpha diversity. And the most representative phyla were the proteobacteria, actinobacteria, funcomycetes, and bacteroidetes. Regarding the structure of these communities, we saw that both types of inocula has affected the structure of the bacterial community, special or in particular in the presence of biochar. Regarding the fungal communities, we saw that the presence of DMF inocula decreased the diversity of rhizospheric fungal communities, and also in treatments with biochar, we observed that the diversity of the fungal communities was lower when we applied the two types of inocula. And the most abundant phyla obtained here was Ascomycota and Basidiomycota. And in terms of structure, we saw that TMF inocula also affected the structure of grapevine rhizosphere fungal communities. So, as I told you, this is ongoing work. So we saw that we have a good diversity in the rhizosphere-associated microbial communities. In general, both microbial inoculants and biochar influenced the structure and also the diversity of these communities. But we are, of course, also still running some tests in order and analysis to see if we have other type of correlations with environmental and also with plant nutritional parameters, mast attributes. So all of these are still ongoing. So I would like to thank, of course, the COST and also the RECRO project, all partners involved in national funding agencies, and thank you for your attention.
We have time for one question.
I have a question regarding the increased concentration that you have observed in the grape.
Mm-hmm.
So do you have data also of the, I don't know, total yield of those grapes?
Yes, we also have those.
And this is comparable. So it's not that you have less water.
We not observed an increase in the production, but we had these results in the quality. We are still trying to understand the impact in the wine quality. Okay, thank you.
Next speaker is Agnes.
Hello, thanks for having me, inviting me here. Oh, sorry. Thank you for inviting me here. I'm Agnès Ardanouille and I work at the INRAE in Toulouse, and I'm going to present work on molecular tools to describe AMF with interactions. And I will see that increase in sulfophosphate availability shifts AMF communities and mycorrhizal nitrogen uptake. Yeah, thanks. This is our team, the team Arbuscular Mycorrhizal Symbiosis Efficacy, and the work I'm going to present has been mainly done by Margot Trinquier as a part of her PhD thesis. So as we all know, arbuscular mycorrhiza interaction has great potential to alleviate reliance on synthetic fertilizers in agriculture, among many other benefits. However, the main barrier for its adoption in conservation in agriculture is the lack of predictability of plant performance. And that we can also call context dependency. So our point in the team is that there's a need to study the nutritional AM interaction in realistic conditions. Like I mentioned, it's a dynamic interaction modulated by the biotic conditions and plant requirements. AM plant can acquire nutrients via the direct plant pathway through the hair roots, and can also acquire nutrients via the mycorrhiza indirect pathway, the mycorrhizal pathway. However, there's an open question on how important is the mycorrhizal uptake pathway for plant nutrition in front of the root acquisition pathway, and for which nutrients is it important. And we believe that there's a need to develop tools to characterize. So here we focus on the potential of periarbuscular membrane transporters as markers of mycorrhizal functioning. Here I present there's a very nice picture of an arbuscule, and the interface between the fungus in that arbuscule and the plant is the periarbuscular membrane. So we ask ourselves the questions: can this periarbuscular transporter serve as marker of plant mycorrhizal uptake? So we want to study a marker of AM symbiosis, that's not a transporter, ammonia transporter, a proton pump, nitrate high affinity transporter, and a phosphate transporter, among others. So for that, I'm going to present briefly what we did. In the first place, we did a validation of the proposed periarbuscular markers in control conditions. Then, as a proof of concept, we used a 50-year-old fertilization assay in field conditions, where we characterized the AMF cumulatives in wheat roots along the P gradient. And also, we used these markers to study the wheat dependence on the mycorrhizal nutrient pathway with these periarbuscular markers. And finally, I'll present a brief complementary experiment in control conditions that help us explain some of our field results. So first, yes, the periarbuscular membrane transporters that we chose are specific of AM. So there's the relative expression of the different marker genes, and we can see in blue that all those inoculated with Rhizophagos irregularis, they present a marked expression in front of the control plants, non-inoculated, that are in yellow. So we thought we can use these tools in our field experiment. Then I will briefly present the field case study. It's a 50-year phosphorus fertilization assay. It was established in 1968 at the INRAE and followed by the Agroécologie et Phénotypage des Cultures, an experimental unit. It consists of a split plot design with randomized blocks arranged along two lines. And there's 4 different treatments. Here I will only refer to 3 of them. It's called P0, so there has been no phosphorus addition since 1968. Then P1, phosphorus addition has been the replacement of P exports each year. And P4, 3-fold or 4-fold at some point of the P1 fertilization. This led, as we can see to this graph, that in 2021, all the different treatments had P0 was around 5 milligrams per kilogram of soil of P. Olsen, and then P1 had 2-fold the amount of P0, and our treatment P4 had 10-fold the amount of P0. So I briefly will present the characterization of these treatments done by Margot with ITS2 fragment with the Illumina MySec. And what we can see in these graphs, we see the relative abundance at the level of genus. And we can see 2 main things. The first one is like as we go from P0 to P4, we see that the purple increases. So we observe an increase of the genus Funeliformis with the increased phosphorus availability. Also, I want to highlight an important thing for me. That's in gray. We can see that in the treatment P0 and P1, but especially in P0, there's a lot of AMF that are classified as an AMF, but they are unidentified even after manual annotation. So I don't want to comment much on it, but it's an open question. I would love to discuss about this further. So then, in the same field trial, we wanted to check if one of our markers, the AM3 marker of mycorrhiza, could serve as proxy of IMF colonization. And we can see that this is the case, but it was not the case when we tried to correlate colonization with 18S copy number. However, we need to double check and still refine this, because we didn't observe in our data set there was no colonizations lower than 40%. So we need to develop these tools further. Next, I'm going to present the same transporters that we already validated, and we tried them with our field samples. And you can see this is graphs of relative expression, and only the AM3 marker, colonization proxy, the ammonia transporter, and the phosphate transporter. And we can see that in the two field trials in 2019 and 2022, we have a contrasting expression of NMP transporters. We can see that in 2019, the first line, there's a maximum colonization in treatment P4. And the opposite is true for the year 2022. Also, when we see the ammonia transporter and the phosphate transporter, we can see that for 2019, in correlation with the expression of AM3, we also have a higher ammonia transporter expression. And the same holds true for the treatment P0 in 2022. So we didn't get analysis of soils at that point. But we have the agronomic trajectories following the fields for the rotations. They are very well kept by the experimental unit. So we know that the yield was different for the whole experimental system in 2019 and 2022, and that the precedent crop was in one case sunflower, and in the second case soybean. So for 2019, we had the precedent of sunflower, and for 2022 of soybean, which we hypothesize might have influenced the soil and that the nitrogen pool was slightly different in both years. To confirm this, we will do analysis of nitrogen content in roots, but this is ongoing. So our hypothesis is that this different nitrogen content might modulate colonization and the relative expression of the transporters. So in order to prove that, we collected soil from the field, from treatment P4 and for treatment P0, and we set up an experiment in controlled conditions in which we added nitrogen fertilization. So then we saw that the results of this experiment validated what our hypothesis of what might have happened in field conditions, and that with reliance on the mycorrhizal pathway probably depends on the interaction, as we know, of nitrogen and phosphorus in the soil. So we could see in the control experiment that there was a high colonization proxy in P0 in both treatments, and in P0 with a high in P4, but that was not the case once both nutrients were highly abundant. So the conclusions of this work and the way forward, we saw that long-term pea fertilization decreased evenness in AMF communities and promoted the Funeliforme genus. Also, the AIM3 expression is a promising marker of AMF colonization in wheat roots, but not the 18S copy number, and that the expression of transporters across the pea gradient was dependent on the sampling year, and possibly due to the interaction of phosphorus and nitrogen availability, and that the expression of nutrient transporters in the periarbuscular membrane might indicate the plant reliance on the mycorrhizal pathway. So we plan on validating these markers with controlling, for example, nutrient fluxes. And I would be really happy to talk about can we further validate these tools to capture the this dynamic interaction in field conditions. Thank you very much.
We will directly go to the next presentation, which is by Gulten. Gulten.
Hello, I am Gülden Balcı from Turkey. I would like to briefly summarize the effect of mycorrhizae applied to strawberry exposed to high pH stress. There are many studies showing that mycorrhizae applied to strawberry improves yield and quality. This study aimed to determine the effect mycorrhizae applied on strawberry in high pH condition of the vegetative growth, biochemical characters, and mineral element intake. In our experiment, the Albion cultivars was used. The experiment was carried out by filling the peat and pearl mixture at the right 1-on-1 in the 5-liter pots. Lime mediation was not done to the pots in control, and 1% lime was added to group to which lime stress was applied to term of weight. In the initial and final pH of the cultivated media in the experiment determined as 7.7 and 7.4 respectively. Perfect in combination powder form contained mycorrhiza at the rate of 23%, named endo-root soluble and belong to BioGlobal firm. You can see mycorrhiza strain in mix in slide. Mycorrhiza application to seedling was performed by soaking plant roots in the solution, which was perfect. I mixed the packet of 250 gram powder with the 10-liter sugar water for an hour before planting. The remaining solution was poured in the plant root area as sub. The plant is in group control group control group were kept in the water for an hour. Leaf samples was performed 3 different periods. The first leaf sample was taken in the period when the strawberry seedling had 4 leaves. The second sample was taken 2 months after planning, and the last sample was taken 3 months after the planning. In the slide, the generation pictures from our study. A brief look at the effects of mycorrhiza on vegetative parameters. Lipharia is reduced under stress conditions. Mycorrhiza application seems to reduce the decline. Show. If we examine some mineral uptake, when biochemical abstracts are investigated, the mycorrhiza application increased the amount of proline during flowering and harvest period. It also observed that mycorrhiza application reduced cell damage during flowering and harvest period. If we examine some mineral uptake of mycorrhiza application in strawberry leaf, stress conditions all at the beginning of flowering and during flowering period are particularly supported by iron uptake. Again, under stress conditions, mycorrhiza application supported phosphorus in all 3 periods. If we examine the mineral uptake, element uptake in the strawberry stems, we see that iron and phosphorus uptake in the stem as well as in leaf is supported in stress condition. Finally, when we look at the mineral uptake, mineral element content of roots, again in all 3 periods, iron intake increased with mycorrhiza use. The use of mycorrhiza increased phosphorus uptake in all periods. Soil pH caused single problems in strawberry cultivation. It was determined that the amount Lipharia increased. Under stress condition, mycorrhiza usually has positive effect on vegetative growth and some biochemical properties. This situation is also detected in iron and phosphorus intake and amount of proline. Use the mycorrhiza can be recommended for use the negative effect the strawberry cultivation in lime soils far from the sea. Thank you.
Okay, so I think we have no time for questions. And we move to the last working group. The working group 4. So with us, an introduction of the objectives of the working group. So I don't need to share the screen now? Duplicate, no? Nobody complains.
Okay, thank you. Here I am once again, this time to talk about Working Group 4, which more or less as Working Group 3 is taking us from the lab to the field. So this working group is called Managing and Improving Beneficial Interactions Networks under Field Conditions. And it's really important because all we've been speaking only makes sense if we can transfer this to field and transform it in real gains for crop production. The main objective is to facilitate the implementation of scientifically well-supported strategies for improvement of beneficial root microorganisms, and also concerning with introduction of beneficial root-associated microorganisms in agriculture as innocuous. So not only managing native populations in the field, and the strategies are very diverse, as we heard for some of the presentations today. So whenever we choose a crop sequence, whenever we choose a kind of fertilizer use, we are also influencing these populations and how can we take the best advantage of them. But also thinking about the introduction of commercial inoculum can be a way. I would like to stress what Jan also stressed. Regarding mycorrhiza, we always keep the focus on nutritional importance, although more and more the importance on their protection against biotic and abiotic stresses is becoming greater and greater. Because in fact, regarding nutritional problems, we have tools to deal with. Regarding abiotic and biotic stresses, we have less and less tools to deal with. So if we can make the best use possible of these beneficial interactions to overcome these stresses that we are facing more and more, is definitely a way forward. So the main objectives of this working group is to identify the barriers limiting the use of beneficial root-associated microbes under field conditions. So if we do know about all these good things these microbes can do for crops, why don't we use them more? What are the knowledge gaps? What are the knowledge transfer gaps? So what's missing between what we develop in our labs, what we know, and the end user, which is finally the farmer. So this is a challenging objective, as it is the objective number 2, which is to propose recommendations on beneficial root-associated microbes, inoculant properties and quality. We do know there is a lot of inconsistency, help me, thank you, in defining the quality and the properties of these products, because they are not very well standardized methods, rules, regulation, European and not only, is not very specific. So this is also what is a good inoculum. This is also how to define it. The other 2 objectives are related to the proposed methodologies to evaluate the effect of these root-associated microorganisms under field conditions, which raises many questions, because under field conditions we have so many more interactions taking place. So what is good in our lab or in our pot trials will be not necessarily good under field conditions, and how can we manage this and get to know more about this. And last objective is to propose strategies for co-selection of plants and microorganisms. Also, we've heard some presentations today that go on this sense. So how breeding systems have to take into account also the presence of these organisms, and how can we promote their action, the ones that go in the seed, the type of varieties that can make good associations or not. So a lot of things are coming in that we are getting to know more and more, and clearly breeding have to take this in the equation of decision making also. We have quite a few deliverables, some from the coming times, namely to have a comprehensive analysis of barriers and key challenges for field use of beneficial associated microbes. So why
why don't we use them more if they are so good and they perform such important things, not only for the crops but for the system and even economically, as Jan mentioned. Recommendations of inoculum properties for quality, so define criteria to say what is a good inoculum, what is a good inoculum or what it isn't, which is very challenging. Have a list of European researchers and laboratories offering expertise and facilities for characterization the potential of these organisms. So if you want to know more about them, where or whom can we address? And deliverable 6 gets much more in work groups 3, but I would say we can, group 4 also is a bit concerned about this, how can we list the services? So in order to convince at the limit farmers, we have to know exactly to explain what are the benefits they can get from this. So this working group apparently has the interest of a lot of participants, around 157, more than 50% of the participants, and the number of countries within the 43 countries, I believe 31 are interested or want to collaborate or develop research in this working group. I invite you to meet tomorrow for the session of discussion in the room, which I also don't know which one will be, to have a kind of a brainstorm to highlight how can we achieve these deliverables by discussing, collaborating among us. So hope to see you tomorrow and take this further. Thank you.
Thank you. So now we can move to the scientific presentation. So there will be first Anna-Maria Pietilä from Finland. No? Ah yes, we can come.
So thank you for having me give this talk here. So I'm Anna-Maria Pietilä and I come from the University of Oulu, Finland. So, and my talk is about endosymbionts as potential tools for sustainable agriculture. So, have you ever thought about how everything in nature works perfectly? Like there's no garbage, no landfills, no roads, no buildings. There might be some structures, but they eventually disappear just by themselves with time. So the way I see it, that nature works perfectly, and we really can learn a lot just by observing and studying nature. So if we think about the countryside, the farmlands, it's what we usually think as nature, but in reality it's just really far from nature, because we have these huge monocultures that we dump with chemical fertilizers that cause eutrophication, and then we dump them with pesticides that create the health risks for ourselves and also kill the pollinators. So it's not really nature. Organic farming is a bit more closer towards nature. There we have these biofertilizers that you all know to replace the synthetic ones. They are the microorganisms that produce phytohormones or fix nitrogen or solubilize the phosphate in the soil. And then we have the biocontrol agents that fight the plant diseases through induced resistance, production of antibiotics or cytophores. And these are mainly rhizobacteria and mycorrhizal fungi. But the problem, as mentioned with these, is usually the inconsistent field performance. So we really need new tools And how to find them? Well, endophytes could represent one possibility. They have the same mechanisms, more or less, as rhizobacteria or mycorrhizal fungi. But the majority of endophytes are kind of overlapping with the rhizobacteria. And they are also studied in the croplands, in the roots of croplands. So they are quite well already known. So how to find something completely new? Excuse me. Our approach has been to look for plants of extreme environments, and we also suggest that micro-rich environments should be looked for, and also not just focus on the already known traits, but really look for something completely new. So for this, we have been focusing on the trees of the subarctic forests, and especially this tree, the Scots pine. And we have been looking for microbes in the shoot tissues, especially in the buds of the tree. And in this way, we have discovered this bacterium, Methylorubrum extorquens. And we have focused our research on this bacterium lately. So we have found that it can really promote the growth of the host, the Scots pine seedlings. In vitro, it induces the growth. We get longer roots and higher shoot biomass, as you can see here. And compared to mycorrhizal fungi, it's at the same level of growth promotion, like you can see in this picture. So here the SV is the Zoilus variegatus, the mycorrhizal fungus, and another is Physolithus tinctorius. So the Methylorubrum is MB here, and it induces the growth of shoots and roots at the same level as these fungi. And it's doing it without producing any known plant hormones, as far as we know. So we got interested in this bacterium, and we studied the colonization process of the bacterium in the pine, and we found that it colonizes the host without breaching the cell walls, and it produces these infection thread-like structures during the colonization. This is from stem. And also infection pocket-like structures that you can see here, the green ones. And also, once it has entered the tissue, it spreads throughout the whole plant through the xylem, and it produces this systemic colonization. So basically, it's using these rhizobia-like infection mechanisms to colonize the host. And not just that, but it's also living inside the pine cells, as you can see in this picture, and even near the nucleus. So we often see the bacterial cells aggregating around the nuclei. So of course, this raises new questions like how and why. How does it get there, and why does it accumulate near the nuclei? And I will focus on this question here. So we next sequenced the genome of the bacterium, and we found in the genome these 22 eukaryote-like proteins. And we formed this hypothesis that maybe this bacterium is producing these eukaryote-like proteins to manipulate the plant nucleus directly. And we studied them and selected one of them to study, to study in more detail this ankyrin repeat protein 2, ANC2 in short. And we did this yeast-2 hybrid screening with it, and we found that the majority of the plant proteins that are targets of this ankyrin 2, the bacterial ankyrin 2, are located in the cytoplasm and nucleus of the plant, and they are mainly functioning in plant development and photosynthesis and stress responses. And we also saw this same result using this study of interaction networks. So we believe that this ankyrin 2 mechanism is related to these plant processes. And next, we made a deletion mutant with the ankyrin 2 protein deleted from the bacterial genome. And here you can see on the left the result with the wild type, so it's the normal colonization throughout the tissues. And then with the ankyrin deletion strain, it could not really colonize the plant. It was just mainly on the surfaces. So that would mean that because of loss of the ankyrin 2, the endosymbiont lost the colonization capacity of the plant. And we also tested whether the ankyrin 2 mutant can still induce the growth of the host plant in vitro. And in this picture, these are the root data, but we also have a similar data for the shoots. I'm not showing it because of time. But root data here, so you can see here the normal wild type induces the growth as before. And then the water control is water control, and then the ankyrin 2 mutant even inhibits the root growth. So this would mean that the ankyrin 2 is involved in this growth promotion response, and it's not through any secreted hormones or other similar compounds, but this ankyrin 2 is in the key role in this growth induction. And we have also another endosymbiont from the pine buds, Pseudomonas xantha, and we have been studying the genome of that one too, and it also has these ankyrin repeat proteins in the genome, so it probably has the same mechanism. And we have also tested this in other, in other hosts, so not just the native host, Scots pine, but we also tested this in St. Paulia and Arabidopsis lyrata and strawberry different varieties. And we have seen that it really can also colonize these plants intracellularly, so they are really endosymbionts in different plant species also. And to summarize my talk, nature provides insights into perfection. I think we can learn a lot from just by observing and studying nature. And by this thought, we have found these endosymbionts from pine, which represent a new type of plant symbiosis. They have this nuclear interaction with the host plant, and they have this new trait, new mechanism with this ankyrin 2 protein that they use to manipulate the cell. So we believe that it can be a new tool for agriculture and a new trait that we have discovered. And here's my research group, and I like to point out these two people who have mainly done this work, Janne Koskimäki and Namrata Parwa, who just had her PhD defense 2 weeks ago, and our collaborator, Caroline Frank, who came up with these eukaryote-like proteins, and the rest of my group. And then I would like to thank you and my funder.
Okay, thanks. There may be time for one question. Yes?
I'm just wondering how easy or hard it is to transform pine. Have you tried to put this ankyrin protein in the plant or overexpress this target?
No, this is all we have done so far. So it was really, really laborious to get all this data done. So yeah, maybe in the future, if we get more money.
Yeah, third question. So, do you know if this interaction is changed by the environment? So, for example, nitrogen, phosphate, heat?
No, we just made this like basic tests first, so we haven't tested in different conditions yet.
Thanks a lot.
Thank you.
So we go on with the next presentation. So this is the presentation by Mika Tarka. And you from that.
first of all, many thanks for the organizers for the opportunity to present our work here. I would like to tell you today about our project, how we found out how to construct a barley synthetic community, which is drought tolerant. In my group, we are working on rhizosphere benefits of both bacteria and fungi. We are working on the rhizosphere plant growth promoting bacteria and plant drought stress tolerance. We have a project on arbuscular mycorrhizal fungi and nutrient stoichiometry. And we also have a project on ectomycorrhizal fungi and the adaptation of oak to drought. And later we also work with maize, root gene expression responses to drought and soil compaction. And today I'd like to tell about rhizosphere bacteria. And our story starts from this research platform, which is from our institute, Helmholtz Center for Environmental Research in Halle in Germany. And this infrastructure it has been implemented to analyze the interactions between land use and climate change. It's based on 80-year prognosis on climate change, which should happen in the mid-Germany so that we do have more rain in the winter, but in the growing season, less of that. We already had a very severe drought periods, for instance, in the growing seasons of 18 and 19. And I would like to today look at the organic farming and conventional farming plots of the GCEF. Claudia Breitkreuz, in her PhD thesis, first made a culture collection from the GCEF grown wheat and barley. And here are the data from the wheat collection. From the rhizosphere bacteria, what you can see are two growth stages, stem elongation and the seed formation phases. And what you can clearly see is already that the pseudomonads are dominant in the culture collection in the early growth phase, whereas they are then subsided by phylobacteria in the later growth phases. So it seems that at least the growth stage seems to have a very strong impact. Other factors which were important were also the organic or conventional farming, and we also had some aspects from the climate treatment, so shown here as A and F, ambient and future climate. And if we look then on the far right, you can see the phosphate solubilization activities. We had a few strains, for instance, from pseudomonads, where they had a very strong in vitro activities, and others which were of lower activity, like the Phylobacterium candidates. But we then moved on also to look at the metabarcoding level, what was happening to the communities during the growth stages, by the climate treatment and by the farming system. And we could see that the most important changes were due to the growth phases, and we had also significant changes by farming system and the climate treatment. So we had kind of a basic information about this multifactorial experiment at the DNA level and the culture collections from wheat and barley. And moving forward to that, we were then also implementing a pot experiment to have a more severe drought stress, mimicking the 2018 and 2019 droughts. So we had now another data set also for metabarcoding, where we could see the enrichment of different bacteria in barley and wheat rhizospheres. And what we now did was that we started the selection of our cultivated bacteria. And we did it so that we compared the bacterial 16S rDNA sequences with those from the amplicon datasets, and the ones which were upregulated by drought or the climate treatment in the GCEF were selected. And then from the culture collection, we then had several candidates. And then we had a second stage of selection, and this took place by test by osmotic stress. Here we used polyethylene glycol, and we had like a minus 1.1 megapascal osmotic stress, which is like in a dry soil. So the bacteria had to also grow in that kind of conditions. And in that way, we could have two different drought-tolerant synthetic communities, one from barley and one from wheat. And now, if you look at these lists, you can see that they are quite comparable from the phylogenetic resolution. We have actinobacteria, proteobacteria, and firmicutes in both of these bacterial consortia. Then Linda Rigethe came and started her PhD project, and she's focusing on the barley drought-tolerant synthetic community. We have the host, barley golden promise. She's working on interactions between the community members. She has treatments with water stress, temperature stress, and we are also working on 2 different soil types. The methods are omics methods mainly, and then also different assays inoculating with plants. If we look first of all the performance of the barley drought-tolerant synthetic community, we could see that it increases the growth of barley, especially during drought. In this picture you can see first of all the phenotype, so you see that the barley inoculated with the synthetic community has not such wilting systems as the control with mock treatment. And what the Syncom promotes is leaf length, both in controls and under drought, but shoot height and shoot biomass are only stimulated under drought conditions. We were then also interested whether it has an effect against biotic stress. And this was tested then with powdery mildew, Plumeria graminis, in collaboration with Corinna Vlot-Schuster's group in Bayreuth. And we could see that when we look at the infection of the leaves by the powdery mildew mycelium, we have much less infection when barley Syncom is inoculated to the roots, and also a significant effect by the wheat synthetic community. So this is where the story is now going. So Linda Rigethe is right now doing the omics experiments to trying to understand the plant response, and also from the metatranscriptome from the bacteria, how they are interacting with each other. I was then thinking how our work would be linking to this VP4. And I think one thing is that I feel that these multifactorial field and greenhouse experiments, they could ones that identify drivers that are important if we think about later rhizosphere management. And I see like 2 possible management strategies here. So one is this kind of a Syncom approach, trying to find crop core microbiomes with certain pools of functions, traits that are important, and of course also competence in the rhizosphere. And another one would of course be also the forced shaping, for instance by drought treatments, what we saw earlier, like leaf infections or low levels of nutrients of root and rhizosphere communities. Now I'd like to acknowledge the people who were involved, and I would like to also mediate a thank you from Linda Rigethe, who got the funding from the Root Benefit to visit Davide Bulgarelli's lab in a few months' time. And thank you for your attention.
For the ESR, so the mock inoculated is no bacteria at all?
No.
Or a Syncom from, I don't know, another plant that is not barley and wheat?
We haven't yet tested another Syncom. It's been no bacteria at all thus far yet.
So the last presentation will be at distance. Get the break before.
Slower. 2 days, that's it.
Yes, I can.
Can you share your screen?
Yes. You see it?
Yes, I need to put it the full screen here. Here. Yeah, I think it's okay. I hope we can hear you well here.
Okay. Do you also see me? I don't see anything, only my screen.
We see your screen for sure. Can you hear? No? Yes, okay, so you can go.
Okay, thank you very much. Hello everybody, thank you very much that I could do this not in vivo, but also not in vitro, but through the screen. I'm Judith van Dingeren, I'm a postdoc in the group of Sophie Goormachtig from the VIB University of Ghent Centrum of Plant Systems Biology. And in Sophie's group, we are studying different plant microbe interaction, beneficial root interactions. So AMF, that was nicely introduced by Naomi. Also PGPRs on different crops, and I will talk about nodulation in soybean. So I will talk about this large citizen science project that we currently finalized, where we wanted to catch rhizobia to introduce high protein containing soybean for sustainable agriculture in Europe. So this is a citizen science project where we did application-driven fundamental research, and where we included the citizens to increase their public awareness of the power of microbes, but also not only for that, we also included really them in phenotyping our plants, in trapping the soybean plants, and also in questionnaires, in interactive demonstrations and discussions to increase the public awareness of the power of microbes. So the challenges that we wanted to tackle was, of course, the ones that we all want to tackle and that fall in this European Green Deal, is to lower the use of chemical and organic fertilizers, to shift our diet to a plant-based protein instead of animal proteins. And how we wanted to tackle this is to increase the local soybean production in Belgium, but in general at northern latitudes in Europe. So you can do that on different ways. So first of all, you of course need to adapt your soybean to grow on these cooler temperatures. So there's a lot of initiatives to breed new varieties, early maturing soybeans. And this is done by a recent VIB spin-off, Botialis. But if you have these early maturing soybeans that can really grow nicely in the northern latitudes, you also need this good inoculum, so this Bradyrhizobium, that results in a nice yield. And the inoculums that are currently used in the fields in Europe are the commercial inoculums that are not really adapted to our environment. So they result in really not reproducible and variable results in terms of protein content. So the protein content needs to reach a certain threshold to allow for nice processing in the industry. So this is not really reproducible with the commercial inoculants. And of course, if you want to do all of this, you also need to take into account your stakeholders, your final end users, your farmers and your citizens. And with this, all of this and taking into account, I would statement that everything is everywhere but the environment selects. We wanted to tackle this by finding indigenous rhizobia that can nodulate soybean, and we wanted to find these in poor soils throughout Belgium, and for that we used the participation of citizens. So we included more than 1,000 citizens, and we asked them to grow soybean in their gardens. And finally, they were also involved in phenotyping these soybean plants, and then they gave back the soybean plants to us, and we checked then if they had nodules or not. Throughout the whole 3 years project, they were involved in this phenotyping in the beginning, but they then were also involved in these different questionnaires and workshops that I talked about earlier. After that, so we got phenotyping data of the citizens. We also did ourselves some phenotyping when the plants arrived in the lab. And we also obtained some soil samples from them, so from all citizens, where we did a physical-chemical analysis. So we measured all the nutrients that were there, and also did a soil microbiome on bacteria, 16S, and on fungi with ETS. And a fungal activity assay, so PLFR. Besides that, we did a big nodule phenotyping, so we have a lot of nodule data. We phenotyped the nodules, we checked how many nodules the plants had, how many plants had nodules, if the nodules were mature or immature. We checked their nitrogen fixation efficiency and the nodule microbiomes. So in total, we only got 34 red nodules, which I say only, but is also still very good. And we also had a lot of white nodules and nodule-like structures. So in parallel, we immediately started with a very large bacterial isolation campaign that you can see on the right here. And we have now a very large collection of bacteria, from which we also have 18 Bradyrhizobium, in which we immediately went to final experiments in the field and in the greenhouse. So this is what you can see here. So we checked 18 Bradyrhizobium in the field if they could perform well on field conditions, and also if they had a nitrogen fixation capacity. So all this data is now captured, and now we are in an ongoing predictive modeling analysis where we want to really link all the soil data, the nodule data, but also so all the data that we have, to link it all together to see if we can have a sort of predictive modeling of to make tailor-made rhizobia, that a certain environment, a certain soil with a certain nutrient condition can predict which bacteria are there, and if these bacteria then will result in mature fixing nodules. So we did already 2 consecutive field trials with these strains that we got, and we could see that here you can see that there are already 3 strains that performed equal to better than the commercial inoculants. This is just based on greenness of the plant, so on chlorophyll content, which gives you an indication of the nitrogen content of the plant. But if you put this more in qualitative values, so in yield and protein content, you could see that 2 of these strains really led to a significant increase in yield and in protein content compared to the commercial inoculants. So finally, we tested all of them, all 18 Bradyrhizobium, and we eventually resulted in 4 Bradyrhizobium strains that performed equal to better. But we have this really big collection of bacteria, more than 1,700, that can now be used for research or application research goals to see if these bacteria can be used as novel soybean biologicals, if they can improve soybean, if they can help the Bradyrhizobium to become better. Are they cheaters? Are they helpers? So there's a lot of different research topics that we can tackle with those. And then, of course, with all the other data, we also have different endeavors that we can go for. So I will talk a little bit about the nodule microbiomes, what we did, but we also have the soil microbiomes, where we could really check the soil legacy. So we also tested some of the soils with indigenous legume, pea legume, which is indigenous to Flemish soils in northern Europe. And here we could see that they grow better, they have very more nodules, so the soil is really adapted to the plant crop, and the plant crop really adapts to soil microbiomes. We also have nutrient analysis that we can correlate. So with the nodulation, so expected correlations are like low nitrogen, really resulted in more presence of nodules. And we are currently also finalizing the soil microbiomes. So here we also tested fungal activity, and we made correlations. And what we found here is also that there are a lot of Bradyrhizobiums in the soils, but that they not always result in nodules. So for the nodule microbiomes, what we saw is that the red nodules always resulted in a low diversity. So which is not completely unexpected, as a nodule, a mature nodule, is really a clonal propagation of one very good Bradyrhizobium, and that all the other nodule-like structures or white nodules really had a higher diversity in microbes. If we then looked at these red nodules and what was there, this was then very surprising because of the 34 red nodules. What we saw is that only 10 of them had really an abundant Bradyrhizobium, and all the other ones had a very abundant other bacteria, which was identified as a Stradifera. If we then used the Stradifera in lab experiments and we just inoculated the soybean plants in the lab, we could see that the Stradifera always induces small and white nodules, but they were never really able to fix nitrogen. And this was not surprising if you looked at the genomes, because they didn't have genes that are related to symbiosis. But they were captured from nodules that were red. So if we then looked a little bit more carefully and then just at the other diversity of this red nodules from where we captured the Stradifera, we could see that they were always accompanied with a Bradyrhizobium. So this is something that we are now further studying. If this Bradyrhizobium is then the fixing one and the Stradifera is the cheater or the helper, this is something that we don't know, and is something that we are studying now. Finally, what we also checked was, okay, we have these red nodules where we could find 10 of them that had Bradyrhizobium, but what about the others? And we could see that also white nodules and nodule-like structures had an abundant Bradyrhizobium presence, but only in the 25 nodules of the 1,000 gardens. So these Bradyrhizobium are, of course, also very interesting to study, to see if you can, based on them, make new strains, if they are helpers, if they are needed, or cheaters. So based on that, I have a small summary. So we only found a few nitrogen-fixing bradyrhizobium, while more of the non-nodulating bodies were not fixing. If the nodule is not functional, we see a higher diversity. So all these microbes that are there, are they then just opportunistic microbes that are just occupying this nodule for themselves? And then Tardivaga, although it doesn't have genes involved in symbiosis, is it a cheater or helping the symbiosis? So that's also a relevant question that we want to tackle for research and application. So that was it. So it's of course a very big project with a lot of different partners. And Lena Vlaming, that was there in red, is the one that really was initiating and organizing this project. So also thank to you for listening to me, the last speaker.
So normally we run out of time, but as we cannot talk at the coffee break, if there is one question or two, you can take the opportunity. No quick. Yeah, why you say there is no co-adaptation? We didn't get this. Yeah, you understood my question?
Yeah, so this, I mentioned this because we found a lot of bradyrhizobium in the soil, so in the soil microbiomes, and also in the nodules and nodule-like structures, but they didn't really fix the nodules, so they were not really adapted to go hand in hand with the soybean and to make mature fixing nodules.
Okay. So thanks and for the presentation and for the session and all the speakers. And now we can go to the break or no? There is two more little talks. Surprise. Okay. Well, anyway, thanks for the talk and the session. And bye-bye.
So there is two last points we would like to present and discuss today. First one is about the communication. Calliope is going to present it. And the second one will be about STSM and some STSM and some discussion recommendation also.
So hello everyone. This is going to be very short. Yeah, okay. It's about dissemination and outreach. Of course, it's a European project. We always have to think about this. And what we have been doing these first months in our cost action, we will try to put up the website, which I suppose you have all visited, identify our logo, do the graphic identity of our cost action, and the content still needs a lot of polishing. We haven't been able to work on the content of the website yet, but this will be improved, we think. So on the website, our idea is that you will be able to find everything that we will need to announce or have to deal with this action. We have prepared the graphic identity, so whenever we're going to need to have posters or whatever it's going to be needed, it's going to be there available for you to disseminate the action in the different activities that we are going to be involved as the whole participants in this cost action. And as Benoit mentioned before, Florian has already prepared this one slide that you can take together with you in all your presentations and activities or whatever we as partners of these constructions are involved. Our idea is that all this communication material is going to be available on the website for you to download every time that you need to do so. And if you need to make small changes to make it to adapt to the specific activity that you are going to be involved, you can contact us to make the changes on the content. For example, here we have the first meeting, but maybe it's something different you are going to do to read, and we are going to prepare this for you and give it available for you to disseminate and use it in your activities. So this slide is going to be available as well. The idea is that because we need also to know where the action is disseminated and where the action is advertised, we are going to ask you to fill in first what type of outreach activity or dissemination activity this is going to be, who is going to be doing it and where, so that we have all the data that we need to prepare the report back to the cost action as needed. And we also have prepared a template for your activities, and the type of activity can be anything that you feel fit to talk about the action and advertise the action. This can be a conference or I don't know. We are going to have outreach activities, for example, researchers night, etc. So if you would like to include the logo and the presentation of the cost action, this is going to be very, very important for the final evaluation of our cost action. Okay, and this is needed so that we persuade them to give us money for the next year and the next year and the next year, and increase the budget that we have available. It's important to have the title of the activity, but very important is also to have numbers. So what is the size of the audience that you are advertising the action, and the countries that this is involved? Is it a local event, or is it an international event? Is it etc. And this is it. We don't have social media yet because I'm not very efficient on that, but we are sorting this tomorrow, we hope. And we will have the usual stuff, so that you can also find the root benefit on the social media as well. And this is it for me, I think. Yeah? Thank you.
So there was a slot to introduce you the first STSM call. Jenny was supposed to do it, but she could not attend, so I will just make a short presentation about the first call and the next call. Okay, so the first call was launched, I don't remember, was February or early this year. So there was 9 applications. So there is a selection committee. It's a grant awarding. The grant awarding committee, which is composed of the working group leader plus the grant awarding coordinator, so Jenny. So they made a selection among the 9 applications. 6 were approved for a total of 13 kilo euros. So as I mentioned this morning, we have a total budget for this year of 26,000 euros. So we still have half of the budget for a second call that we are going to launch soon. So I just would like to remind you that this short-term scientific mission grants. It's a contribution to travel, accommodation, and meal expenses. It can be up to 4,000 euros, but we expect application around 2,000 to allow more people to have grants. It does not cover all the expenses you have, so it's a contribution, so there might be a contribution of your own laboratory as well. Importantly, for this year's, the mission need to be done before September, because then you have to write a report, and then you're going to receive the grant after the report only, and we need to finalize the budget or use all the budget before end of October. So that's why end of September is the limit for your travel. So you can start to think about it. The second call will be launched soon. So what are the criteria? Clarity and quality of the application, as always. Contribution to the objective of the action. Also, you have seen the objective of the 4 working groups, so you can explain how your mission is contributing to one of the other of the working group. Feasibility of the working plan and the outputs of the mission. Okay, I think that's all for the STSM.
What is the criteria for to approve the students who are the participants? This is the criteria that you have for approving the STSM?
Yes.
And what is the criteria for this? You choose 6 out of 9 applicants.
Yes.
What were the
criteria?
This criteria. So the
criteria for the students, not for the STSM.
Was it only on the project, or also on the students?
So yes. Another thing we need to keep in mind, in any of the activities, we also have to follow the cost policy. And the cost policy is also a balance between the countries, between the age, and especially the STSM should go in priority for the young researcher. And the last criteria is the gender also. It should be equilibrated. So it's also criteria that we have applied to selecting the participants to this meeting, and especially those we have talked today. And these need to be balanced in the 2 calls. So let's say in the first call we have took it in account to a certain extent, but definitely in the second call we need to make sure that for the first year that will be taken into consideration, and it will be the case each year. But let's say that's come at the second layer after those I have mentioned, like quality and compatibility with the objective of the action. So we also think that it was important to start the first call as soon as possible this year, but we also expect that after we meet during this meeting there will be new opportunities for a second call. Okay, so just to take the opportunity to also introduce what we plan or for next year. So this year there was two meetings, general meeting now and a workshop that's gonna take place in Portugal in September on topics driven by the I don't remember how we call it, the stakeholder-driven topics. So and which will be about inoculation, mainly inoculants. Next year there will be only a general meeting, no workshop. We plan to have a similar workshop on the 3rd year. This year there is an online training school. We hope that next year we can make a face-to-face training school. There is no topic yet defined. And next year they're going to be again the short-term scientific mission grants and the ITC grants. This will come each year. So this is also to tell you that your proposition for teaching school topics, transversal or working group topic that can be discussed all together, or also workshop, they are welcome. Nothing is fixed now for the next year, so we expect you to propose also ideas for the activities for the next year for the the network. And finally, just some important information. Don't forget to sign the attendance list. Have you checked? Yeah, if everyone has signed it?
Yes, I checked.
Okay, so that's a good point. Yes, another very important information, there can be a situation in which you buy a flight ticket but you cannot come. That's the case of Judith today. She was not able to come. If you do not sign the attendance list and you are not here, we cannot reimburse the flight ticket. So it's very important to take a cancellation insurance. It's not very expensive. This can be refunded, the insurance. And it's very critical if you buy a flight ticket or train ticket and you cannot come. So sometimes you have one associated with your credit card. You might already have one, but you can also buy one on top of it. So it does not mean to have a refundable flight ticket. This is often very expensive. It's just an insurance on top of the flight ticket. Sometimes it's just a few tenths of euros. Yes?
So, actually, first of all, if you don't come and you bought a ticket which you can't reimburse by your aviation company, we can't reimburse you. Can't. And this is what happens with Judith, because she bought her ticket and we can't reimburse her even if she could participate. So think about it. The second point, when you take in insurance, so actually we can reimburse just for the cancellation. But if in your insurance is written medical reason and the price of the medical reason, we can't reimburse a medical reason. But if there is written like list of what is this and one price, we can reimburse everything. Okay? That's right? You understand?
Make it clear. Sometimes the insurance, you have just one price, and it covers several things.
Yeah.
Be careful that it's not like several lines with a price for each of the thing which is insured, because the cost is check-in and it's relevant in only cancellation insurance, which could be for medical reason, but I think it's a medical assistance which is not covered. Like, okay, if you have any doubt, just write us an email. But think about taking insurance for next meeting or anything, training school or whatever. Also, one more information about the reimbursement rules. So we can reimburse the long distance travel. And then the daily allowance is covering lunch, dinner, hotel, and also short distance travel. So that's very clear. We cannot reimburse like bus, metro, taxi, which are the short distance. So, and long distance is over 100 kilometers. And all the other, it's covered by daily allowance, which usually is supposed to be enough if your hotel is not too expensive, or the restaurant you have to go. So then, after this meeting and after each activities, you will have to claim refunding on eCost. I think it's quite simple on the website. And then we will access to your claim on eCost, but the reimbursement will be done by INRAE, because the money is not on Cost. Cost has transferred the money to INRAE, and then we are going to make the bank transfer. So that's why Yara asked you to fill a form, because we need to enter each of you in our informatic system to be able to make a bank transfer. So it's a bit like double work, but it's like this in all action. It's not only in our action. That's the principle how the cost action works.
My problem is that it's not safe to send all kinds of bank details in email in this day. So
yeah, we can find other way if there is some risk.
Send it
Yes.
now. Do we need to send it every time?
No. Once you are registered in the system, it's fine. So like all of you who have participated once, you are in the system. Yes.
But you need to know if you change the bank.
Yes. But this is to explain to you why we ask you to fill this form and sign it, because we have to enter you in our system. So actually there is two possibilities where either you have paid by yourself the flight ticket and reimburse it to you, or your institute has paid it and we are going to reimburse your institute. Depends on that. There is the two options. And I think that's all for tonight, or from my side.
No, just one moment. So it's just the first day, but tomorrow will be the second day. But I will ask you to send as soon as possible your climbing, because if there is some trouble, I don't know if there is there's some stuff which I want to precise. When you send to me your transport tickets, for example, I need to know that it will be a name of the company, the date and the hours when you come, when you are going and when you are coming back. If you send to me the information there is no hour, I will ask you and we'll wait for this. So please do I already sent to you the information about the reimbursement. Read again. If you have any question, you can call to me. Call me maybe.
Why hours is important is because the daily allowance is calculated depending on whether you arrive the day before, on evening or morning. So this is to calculate the amount of daily allowance. We need to know exactly at which hour you arrive.
The information that you are requiring, we can fulfill through ECOS system. So I don't know if somebody fulfill it. I don't know where is the need.
Okay, so yes, you will put all of this information to the eCost, and me, I have access to all of this information. So I will look everyone, and if there is some question, I will ask. Okay?
Your claim will be on eCost only. Yes. So you will put everything on eCost.
As I know, it's with asterisk, so it's by default required the hours and the.
Yeah, yes.
So
yes,
you
but
cannot go.
I mean, the cost officer that told us even if eCost is everything is mentioned clearly, it's always some participants are not given the all information. So just to say, now we are going to have some more, one more break before the dinner where we have something to drink, I think, here. There is a poster also. I encourage you to go to the poster. And you have also prepared something for us to, yes, ice-breaking events.
So actually, right now it's the most interesting part of the party, and we will discuss about the posters if someone have a question. But there is already prepared the ice-breaking party, and you're welcome. We're going.